I am Obi Ifechukwu from Nigeria, from Team 6. With my teammate here, Samuel Zagabe, we are here to argue that genetically modified microorganisms should not be widely used to combat antimicrobial resistance. Microbes, bacteria, viruses, fungi, and parasites increasingly resist existing drugs, making common infections hard or impossible to treat. In 2019, drug-resistant infections caused approximately 1.2 million deaths globally. Escalating antimicrobial resistance is a top global crisis, killing millions and raising treatment costs. Scientists propose novel tools, including genetically engineered microorganisms designed to degrade antibiotics or target resistant pathogens. The misuse and overuse of antimicrobials are the main drivers in the development of drug-resistant pathogens. Lack of clean water and sanitation and inadequate infection prevention control measures promote the spread of microbes, some of which are resistant to antimicrobial treatments. Genetically modified microorganisms are deliberately modified bacteria or viruses intended to reduce antimicrobial resistance by breaking down excess antibiotics or killing resistant strains. They are released into the environment or patient is what we are here to debate. Genetic engineering, also a genetic modification process, is the act of using laboratory tools to change the nucleic acid sequence of organisms by removing or adding base pairs, inserting or inactivating an unnecessary virulent gene by creating a new characteristic altogether in genetically modified microorganisms. One of the applications of GMs is their potential activity against resistance to antibiotic bacterial pathogens. Another application of GMs is seen in vaccinology. Vaccines are the main strategy for preventing most infectious diseases. My team is here today to stress the unpredictable impact of GMs in the human microbiome, ecosystems, and safety. We caution that rushing GM deployment might lead to unintended consequences. My team is here today to argue that GM deployment poses serious unknown risks. These engineered microbes, when released into the environment, may behave unpredictably and spread genes beyond control. Our key concern here is that safety is unproven. GGMs could outcompete native microbes or evolve new traits, and inserted genes may transfer to wild organisms. We emphasize precautions. Unintended consequences, that these new strains, could outweigh the potential benefits by using these GGMs. The burden of proof, therefore, lies on showing that GGMs are safe. Given limited ecological data and experience, deploying them widely, which are these GGMs, is premature. Our constructive arguments against the fact that GGMs should not be used to combat AMRs are: one, the uncertain environmental impact and horizontal gene transfer. Engineered strains, engineered strains may not stay contained. Lab modifications may behave differently in complex ecosystems. Studies warn that GGMs could outfit native microbiome or express unforeseen phenotypes in new niches. Crucially, horizontal gene transfer is persuasive. Hence, the use of GGMs to fight AMRs could lead to potential ways or sources through which new strains of bacteria and viruses could be spread across the entire biosphere, risking novel resistant pathogens. Our second argument here is that the current oversight for deliberate use of GGMs release is inadequate. Experts note that regulators have little experience evaluating environmental GGM safety. This regulatory uncertainty creates gaps and delays risk assessment. Moreover, synthetic biology poses dual use threats. Engineered microbes could be repurposed maliciously or accidentally as biothreats. Without robust governance, containment failure may lead to severe global consequences. Releasing GDMs into nature raises profound ethical and societal issues. The public wariness of genetically modified organisms is due to long-term safety and tampering with nature consent. Transparency and consent are lacking. Ecosystems and communities cannot opt in to trials. Research has shown that genetically modified microorganisms, even in agriculture, trigger public distrust, and applying similar technology to microbes in the environment could spark extensive backlash. Ethical debates include responsibility for unknown harms and equitable decision making. I am also here to argue that the prudent use of existing drugs and infection control mechanisms are proven cost-effective measures to slow resistance of AMRs. Investment in new antibiotics and vaccines is critical. WHO reports have shown that there are just very few antibiotics that are used to combat AMRs, therefore highlighting the need for more research and development rather than risky environmental interventions such as the use of GGMs. Therefore, we should focus more on using the existing drugs that we have on how to improve their efficacy and use them to reduce antimicrobial resistance. To beat out. Our opposing team argues that the use of GGMs in AMR is precise or involves precise targeting. However, real-world complexity defeats precision. Lab demonstrated target specificity often breaks down in heterogeneous environments where microbial communities and physiochemical factors widely vary. Engineered recognition of kill switches can misfire or degrade, leading to off-target killing or survival of modified strains with unintended traits. Even a single nucleotide can change or alter the host range or regulatory circuits and transforming a precision microbe into a generalist with unpredictable ecological impact. The opposing team argues that GGMs can overcome resistance. I'm here to argue that GGMs can themselves become resistance hotspots. Engineered resistance degrading enzymes and antimicrobial payloads impose novel selective pressures. Surviving microbes or mobile elements can acquire and spread these new genes via horizontal gene transfer, potentially creating super resistant strains rather than eliminating them. Unlike small molecular antibiotics, once a GGM's mechanism is compromised by mutation, redeploying it safely becomes far more complex. Also, the opposing team argues that the use of GGMs reduces antibiotics dependence. Alternatives are already doing this. I am here to argue that I am here to argue that alternatives are already doing this safely. Phage therapy and CRISP antimicrobials selectively kill pathogens without removing or introducing new genes into the environment. And have reached and these therapies have reached clinical trials with promising safety profiles. Therefore, investing in GGMs divert limited funds from proven stewardships and vaccine development, which are more reliable to reduce antibiotic consumption and AMR burden. Another point the opposing team uses to argue is that the use of GGMs in combating AMR is quite eco-friendly. However, I'm here to argue that ecological disruption risk outweighs green claims. Introducing self-replicating organisms, even intended to degrade pollutants or antibiotics, can upset nature cycles, outcompete keystone species or alter food webs in irreversible ways. Engineering, scaling, and formulating of GGMs, their productions also by fermentation, purification, and encapsulation consumes substantial energy and generates considerable amounts or considerably large amounts of waste and carbon footprint. The opposite team argues that GGMs could combat biofilms. Just as bacteria evolve antibiotic resistance. They can also alter biofilm. GGMs can also alter biofilm composition or produce inhibitors against GGM-derived enzymes or toxins, negating the intervention and potentially selecting for a more robust, harder-to-treat biofilm. Hereby close by saying that there is high uncertainty around the use of GGM, which involves ecological disruption, uncontrolled spread, and new antimicrobial resistance pathways. Therefore, research and development into the use of GGM to combat AMRs involve should should involve rigorous evidence-based risk assessment and strong governance before any of these techniques are used widely. Pending when that could happen, I suggest that we tackle AMR holistically by integrating human, animal, and environmental surveillance. We should also promote stewardship programs and invest in research and development of already existing antimicrobials that are used to combat these microorganisms, that are used to combat pathogens. Also, if you try to build informed consent, build public trust through education and strict legislation or ethical oversight over the use of genetic methods to improve lifestyle. We can also preserve antibiotic efficacy via international stakeholders partnerships, modern genetic engineering techniques for stability, comprehensive environmental risk studies, and judicious antibiotic use. Thank you. These are my references. Thank you very much.